All right, it's 2 p.m., so I'll get started. So today is going to be a little change in the topic. Usually this is about fMRI, and today I thought I would bring you more about anatomical imaging, so purely MRI, structural imaging. And I'll put the focus on imaging the CNS, so not only the brain, but the spinal cord as well, and looking at non-healthy tissues, so basically neurological diseases like MS. So for those who don't know me, I'm a staff scientist at the Translational Neuroradiology Unit, or section now, which is directed by Dr. Reich. He's a neuroradiologist trained at Hopkins, started a lab back in 2008, 2009. I joined as a postdoc at the time. And when he put together the mission statement of the lab, the idea was really to use advanced imaging to better understand the source of disability in MS, but also ways to adapt those techniques in clinical trials as well as in daily patient care. So since then, our lab has been working extensively along those lines, and I thought I would show you guys two examples of our current topic of research that kind of make the point of what we're trying to achieve in the lab. So for those who are not familiar with MS, it's unfortunately a very disabling disease that affects young people in the US. And the prevalence right now is about 400,000 in the US, 2.5 million in the world. Its origin is still unknown. Could be multifactorial, could be some genetic susceptibility, could be environment, could be food, could be many things that have been linked to MS, but there's not been a unique trigger. But the pathology is fairly well understood. So it involves multiple components, such as inflammation, demyelination, axonal loss, ultimately neuronal damage, neuron loss, which is really the disabling part of the disease. If you have MS, you're likely to experience any of those symptoms, and the list is pretty long, unfortunately. And although we have treatment to manage those symptoms, we don't really have any cure for this disease. And this is typically shown in this kind of cartoon that is often shown by clinicians in MS, doing MS research, is that you can see the patient as they move on from their initial diagnosis, probably in the second or third decade of life, will have relapse remaining, so they will feel better about themselves after having an attack. But at some point, the disease burden starts to add up, and then they start having more and more permanent symptoms. Unfortunately, after a decade or two, this relapsing course changes to a really chronic, progressive MS disease course, which is really adding in terms of disability, and patients end up being unable to walk by themselves, being on a wheelchair, and so on and so forth. So it's pretty disabling. It doesn't kill as per se. So the average lifespan of MS patient is a little shorter than normal, but it's not a killing disease in the sense that you have a short-term life expectancy. So there are really two big phases in MS. So I just presented before, the early part of disease, which is relapsing remitting. So you have attacks, then you remit, and so on and so forth. And then there's the secondary progressive phase of disease, which will last until the end. And in the MS research field for drugs, they've been a huge advance in number of treatment available for the patient in the past decade. From 1 or 2 drugs in the 90s, you're up to 15 different options right now if you're affected by this disease. So clinicians have many options to test on the subject. However, all these drugs really target the early part of disease, so these inflammatory processes, the relapsing remitting phase. There is really nothing efficient in the progressive phase of disease, which is really lacking right now in the field. And so it's important, therefore, that people who are affected by this disease are diagnosed as early as possible, because the goal is really to limit the impact on the long term for the patients. And so to diagnose MS, unfortunately, it's not a straightforward process, and it's not a single biomarker, a single test that tells you, okay, that you have MS. Unfortunately, clinicians have to deal with a lot of information, which is partly medical history, partly neurological exam, and then lab tests. Could be MRI, could be blood test, could be EEG, VP, could be all kind of different paraclinical testing to help rule out if first it's not MS, it could be something else. And then confirm the diagnosis himself by following this criteria that has been proposed back in the 2000 and been revised a few times, called the Magnolia criteria, And the goal is really to demonstrate that there's been a dissemination of the CNS lesion in space and time. And so to that, you have a quite complex algorithm that you have to follow. Here's a summary in the table where basically clinician is looking for patients that had at least, for example, 2 attacks. So that's the first line of this table. And they have proof from the clinical exam that these lesions that they see, they can assess by neuroexam exam are actually disseminated in the CNS, so there could be a spinal cord or brain lesion. And if that is the case, then they can quickly come to the diagnosis is actually MS. Now, it might not be that simple, and sometimes you have patients that have 2 attacks, but clinician can only detect 1 asymptomatic lesion, and therefore you need to bring some more evidence that there is dissemination in space. And that's where MRI, for example, is useful because you can look at asymptomatic lesion in MRI. Or you can have an earlier process where you have only 1 attack, so the patient just had a single episode and comes to the neurologist and say, you know, what's going on with me, and you do all the battery of tests. Then the clinical exam shows that there's actually a dissemination in space. There are 2 different lesions probably located in CNS, but then you need to show that it's evolving over the time, so it's dissemination in time. Or you can have the even earlier attack where the clinician was lucky enough to catch the patient very early in the disease. And what you try to prove is by the clinical exam and the MRI there is indeed dissemination in space and time, which has really been helpful to detect earlier and earlier MS in patients with those type of single attacks, so we call the CIS, or clinically isolated syndromes. So MRI is an exquisite tool to detect pathology in the CNS, and here are examples where we illustrate this dissemination in space on the MR. So you see the brain has a lesion pointed by the red arrow. This is a periventricular lesion, so it's located next to the ventricles, where the CSF is, and it's located inside the white matter. You have also a location in the infrasternal area, and you can have also a lesion in the spinal cord, right? So if you can see any of those, you're likely to demonstrate this dissemination in space. To show dissemination in time on the MR, you can either do a follow-up MRI and you see a new lesion that tells you there's activity and there's actually new lesion coming up in the brain, or you can inject a contrast agent in the arm of the patient and then look up a few minutes later on the MR if any of the lesions are taking up the contrast agent. That tells you the lesion is active. It's probably a new lesion that was forming recently, and that allows you to prove dissemination in time in a single MR. You don't need basically a follow-up. So it shortens again this period of diagnosis, which is so crucial to start early on the treatment, and impact and limit as possible the long-term impact of the disease. Unfortunately, these criteria are very sensitive when applied in the right conditions, where the clinician has done a good job at really ruling out any other diseases. Unfortunately, it's not always that simple. There are sometimes very complicated cases, and that leads to misdiagnosis, which is happening actually quite frequently. Recent studies polling clinicians across the US, looking at metadata analysis, have shown that actually it's fairly common, between 5 to 35% to max estimate. That is a lot if you think about the 400,000 people that I was just mentioning in the US that currently probably have a treatment that costs about $80,000 a year, and that's going to have that until the rest of their life if they're not undiagnosed. So when you're talking about healthcare and the money that we're spending on healthcare, this is a big topic that we should work on. In our institute, try to limit the cost and have a better use of drugs, especially in MS population. So if we can reduce misdiagnosis, that will be helpful for the economy, but also for the patient himself. I mean, they can experience pretty severe side effects. So of course, this is also has a huge impact on the life of the patient. So we know that these criteria are imperfect. And that's partly due because although MR is very sensitive, it's not specific. Many other conditions will show lesions in the brain like I show you. Could be migraine, could be fibromyalgia, could be small vessel ischemic disease, could be NMO. And so that's why there's been a regain in interest in looking back at the pathology of the lesion in MS and seeing is there anything really specific about MS that we could image and maybe improve the accuracy of our criteria. So Charcot, who was in Paris, La Pitié Salpêtrière in Paris, was one of the first to report MS. And looking under the microscope at his tissue, he started drawing those cartoons, and he pointed out, if you can read French, which fortunately I can here, I can translate for you guys, it's basically telling that there is actually a small vessel at the center of the plaque of the lesion. And this has actually been known for quite a while, and only recently has been revisited by MR. So there is a paper from the early 2000 by a Dutch group led by Barkov, which is one of the big god in the field of MRI criteria, who actually showed that you can detect that in vivo using a 1.5 Tesla MR, using this sequence called susceptible weighted imaging, which is very sensitive to susceptibility effect in blood, the level of oxygenation in the blood, as you guys probably know from fMRI studies. And so they already demonstrated that indeed you can image by MR this central vein. And of course, with the advance of the technology, higher field strength, stronger magnets, stronger gradients, you can really push the in vivo resolution. This is data that I collected here at the NIH using the 7 Tesla scanner that maybe some of you are scanning on for the fMRI experiment. So it's a patient that we put in the scanner that obviously has MS. And you can see those hyperintense lesion and round shape, and then right through the center of this, you can see this dark line that are running through partially or entirely, which are actually those central veins that Charcot described back in the 19th century. And you pretty much see it anywhere in the brain where this lesion is located. So that has actually stimulated the field. This report is not unique. A lot of people using 7 Tesla imaging a few years ago have seen that, and it's been stimulating a lot of research clinicians to actually look at that more routinely and looking at more clinical scanners, like 3 Tesla scanners. So we have collaborated with somebody who has worked a lot on misdiagnosis at the University of Vermont, and Andrew Solomon, who was the first author on this misdiagnosis paper. And so we've been imaging his cohort with some of the technique we developed in that. And basically what he sees in his MS patient cohort is the same finding that I showed you in MS, in our MS. There's this dark line, dark vein, and then around it there is this either round shape or ovoid shaped lesion that develop around this vessel. But if you look at this migraine patient, he's got a fairly large cohort of this patient at Vermont, you don't see this feature anymore. As you can see, the red arrow point to lesion are about the same distribution as MS lesion, same shape, but it just don't show the central vein. So that attracted a lot of interest from many scientists looking at this issue from other sites. So there's been a collection of Siegel Center studies across the US and across Europe looking at their own cohort of migraine, of NMO, of SUSAC, of small vessel disease that can actually mimic MS on the MRI presentation. And they've all been very consistent on one point, is that there's a stark difference in the percentage of central vein in those conditions versus MS. So in green, I highlighted the percentage that either an average or median percentage that you see central vein in MS lesion. It's fairly high. It's anywhere between 70% to up to 90%. But in red, these are the mimics, and you can see it's actually fairly low. And there is a clear dichotomy, there is a clear separation in those two cohorts, I would say. So a lot of attention has been focused on the idea of maybe we could use this criteria, this central vein as an additional criteria for diagnosing MS. And the fairly young consortium born a few years ago, and Danny, our lab chief, was one of the founders of this consortium, actually addressed this in a consensus statement that I recently helped writing, which basically proposed this following idea. So there is clearly single center data that are very consistent and that show there is a lower proportion of central vein positive lesion in other conditions than MS. But there is clearly need to investigate more of those diseases because this is just a small sample of all the other diseases that you need to rule out in differential diagnosis. And then you need to start thinking how you're going to actually discriminate those populations. Do you do a percentage? Let's say if you're more than 50% of the lesion, then you're MS. If you're less, you're not MS. But what happens if certain patients have more than 50? That might bring some confusion. Or do you start picking lesions? So you say, I'm going to pick certain type of lesions and just look at those lesions, which also will speed up the clinical process, right? Because you can count central vein, all the lesions. Imagine your patient has 40 lesions, how much time the radiologist have. We won't be practical. Or do you think about combining maybe the existing criteria, so the DIT, DIS, with the central vein? So there is clearly a need to address this question, and therefore, having a consortium approach to it, using the power of multi-center studies, becomes critical here. And this is what we've been proposing in the consensus statement, is try to build up cohort through multi-center approach, where we image the patient using similar protocols, but we try to look at as many as possible different diseases. Or you can just look at patients undiagnosed yet. So you want to move prospectively. You want to go in patients you don't know the disease. You're going to follow them over time, and you're going to compare the existing criteria versus a more refined criteria that include the central vein and see if there's any difference. Or you can also look at the ones you think are actually not a mess. Scan them with a central vein sequence, see whether it's true or not that you don't see as many central veins in those patients. So there are many different approaches, but the common ground is here, go for multi-center studies so you get statistical power and really makes meaningful conclusions that can actually impact the clinical field. Now, if you look at the typical MRI protocol that is run in the clinic, wherever you go in the private center or the hospital, it's usually very short. You have about 30 minutes, then you have to bring the next patient, bam, bam, bam. Nothing compared to what we do at NIH, where we scan for an hour and a half. The hospital setting is much more stressful. So this is the bare-bone protocol that is recommended per the consensus statement from the European consortium, the MAGNIMS, which is an older brother to our NAMES consortium in the US. And these people have been, in the field for a long time, and they've put this really carefully thought protocol that includes T1 imaging, T2 imaging to pick up those lesions that I mentioned to you, that we look for the DIT and DIS. And then what they do, typically, they inject the contrast agent because they need to prove the dissemination in time. However, you need to wait for the gas to circulate through the body, reach the brain, penetrate through the blood-brain barrier into the lesion, and then you can image at that time properly so you can really see if there is any enhancing. So there is a minimum wait of 5 minutes. And after that, you can do your post-GAT T1, which really shows up your enhancing lesion. But you think about it, this is just basically a waste of time. And some people have been squeezing research sequences for their own purpose, but you could think about actually using that time to run whatever sequence you want to detect your veins. Could be this T2 star, could be the SWI sequence. And you will not add any extra time, so you're still compatible with all the clinical pressure that you encounter in private centers and clinics. So if you go to your scanner and try to do the standard protocol that Siemens or Philips or GE allows you to do, this is what you will get after some tweaking. In 5 minutes, usually this is what you do. You can do a fairly high plane resolution, so you can start seeing those small venules, small veins where the lesions develop around. But you're going to have to sacrifice on the slice thickness. So if you start reslicing your volume in coronal sagittal sections, it's going to be a little more blurry. It's going to be just harder to detect the veins. And also the contrast is a little funny. So this is not purely two-star weighted sequence. You see that it's like a T1, a little bit of T1 sequence. The CSF is darker. So it's not even a very clear image to look at to detect lesions, basically. So when I came in as a postdoc, I had the opportunity to work with a very talented neurologist called Dr. John Batman at the clinical center, who suggested me to try out a new sequence and play with it on the scanner. So I spent a few weekends, actually a lot of weekends, testing, optimizing, refining the protocol until I reached this 5-minute scan that allows you to do a 0.5 isotropic. So this is 500 micron isotropic on a 3T scanner of the whole brain. And as you can see, it really has a nice contrast, so you can really pick out the blood vessels, but you can also find out the lesions that we're after here in MS. And you can see the central vein running through the lesion in all planes. So because it's isotropic now, you can reslice the brain in any direction you want and track your vessel as a line or as a dot. And you can really convince yourself that it's actually central position inside the lesion. So I took on this project a step further through a great collaboration here at the NIH, through different institutes, my institute, NNDS, NIMH, and the clinical center. And then we talked to Siemens. We have a research agreement with them. And we thought, why don't we just start pushing this more into the product pipeline and see how it goes. So with the help of Siemens engineers, we refined the sequences. And in a nutshell, basically what it does, it's like a slow EPI that you guys do on fMRI. So if you heard about the nuts and bolts about how to acquire an EPI sequence, basically is you try to go through your case space or your MRI data as quick as possible. Typically it's done in a single shot, which allows you this high temporal resolution. What we're trying to do here is just to slow it down, because I don't need to scan my patient in a few seconds. What I need is good image, anatomical image. If you go too fast, usually you have too much artifact. So what we did is we did a segmentation. We acquire a certain number of segments per shot. We don't do the entire case space. So we segment the case space in different shots. That reduces the speed of the sequence, but also reduces the amount of artifact. And so it's just a more efficient way to sample your case space compared to the previous sequence, the product sequence I showed you guys initially, the SWI type of sequence. And here's what happened when I put it on different scanners. I don't know if you can see it very well from here, but anyway, we have tried 1.5, 3T, 7T, so we try all across the systems that we have here at the NIH in B1, and they all come down to about a 5-minute protocol. And you can do this sub-millimeter imaging of the whole brain in about 5 minutes or less. And this is the result that we get again on the Siemens scanner at 3 Tesla. And again, this is an MS case, and I can show you that you can basically detect this vein and a lesion in any plane you want, which is really a plus for radiology. So just a final example, here's the product sequence, here's the prototype sequence we developed at NIH, same scan time, you see the contrast is very different, and obviously much easier to pick at the veins on our sequence on the right. And then of course when you look at the coverage, which is crucial in MS because you want to make sure there are lesions in the suprapatatorial brain, but also in the infrapatatorial brain. So this is important here, and you clearly the prototype sequence destroyed the product sequence. So you can do many things also with this sequence. If you're not especially interested in central vein, you can look at other things like vasculature of the brain. If you do a MINIP, this is an example of reconstruction of these images just to highlight the tree, the vasculature tree of the brain. Or you can do this more advanced sequence processing called QSM that look at the local susceptibility of the brain, which became very fashionable right now on the topic of iron accumulation in the brain, how to measure that in vivo. This is just another little slide to show you the benefit of acquiring this sequence during injection. Remember the protocol, that standard MS protocol, has this 5-minute delay time that we want to use to run this sequence. And interestingly, having GAD on board actually helped us, because when the gadolinium is still inside the blood pools, so circulating through all the venules and vessels, blood vessels in the brain, it brings more susceptibility effect, which make these blood vessels more apparent on our scan. This is what I'm trying to show you with the example in the middle. This is my API acquired during an injection of gadolinium. So we push the GAD either manually or with a power injector, and for the next 5 minutes we acquire the scan. And this is where the GAD hasn't penetrated yet the brain and it's still in blood pool, and really brings this increased detection of blood vessels. So it's all benefits for us to actually acquire during the injection. And finally, to show the applicability of the sequence in all kinds of scanners, here's an example of one MS patient that I scan across different platforms. So this is going from 1.5 Tesla magnet that you will find in private centers. If you walk out the street and go to Bethesda, MRI centers have that machine, and you can actually do this kind of thing and still see your blood vessel. If you go to Hopkins or the more fancier hospital, they're going to have the 3T, and you can still run the sequence and see your vein. If you're really top of the line research and have access to the NIH, this is what we're going to see here in your brain if you have an MS lesion. So this sequence works on all kinds of platforms. And it costs manufacturers too. Like I say, so I started working on Philips scanners with Dr. Batman at the radiology department, but then we developed this relationship with Siemens, and if you scan your patient on one or the other, it doesn't really matter. The detection is the same, which is really important if you want to move out this thing into the clinic world where there is no preference for scanners, right? So this is just putting that in numbers, but basically there is a very high reproducibility in detection across scanners. So for all these features, of course, we have encountered a great interest in the community, and already this protocol has been dispatched across more than 15 sites outside the NIH. And we're pushing this, hoping that clinicians will take it up and demonstrate that indeed in their clinic, they can actually replace that dead time with this sequence and get some benefit, some important information about the diagnosis process. Now, if you are a clinician, this is in kind of new contrast. It's very rich. There's a lot of information, a lot of heterogeneity as well, and it's kind of hard to interpret. And clinicians, when they look at the case, they have a few minutes to make a call to how many lesions do they see, how big lesions are. This is way too complicated for them to analyze, and they're not trained for that. Clinicians are used to look at that. That's called a flare. It's a cleaner image. The CSF is dark. The brain, normal brain is gray, and the lesion pops, they're bright. So it's very easy to quickly assess the brain and make a report. So what we thought a few years ago is, why don't we combine those two in one? Take the benefits of both sequences so that the clinician doesn't have to retrain himself and can just look quickly at the images and still have the information about lesion load, but then on top of that, look at the veins. So you see the flare star, that's what we coined this technique, the term we use for this technique, has a lot of features as a flare, so it's fairly easy to pick out the lesion quickly, and then inside the lesion you can see a central vein that is usually picked by the 3D PI sequence. And if you combine in a smart way, you can actually retain the high resolution and isotropic features of the 3D PI on your flare star. Therefore, if you cut it in all planes, you can again see this beautiful little vein inside the lesion in all directions you're looking at to convince yourself this is actually a central vein. So there's the pipeline. There is no magic here. It's very simple, which sometimes is the best. Simple things are actually easiest to carry over to the industry. So you need just a basic rigid concentration of the two volume. You have to interpolate because they're not the same resolution. And usually this is done on the flare. So we upsample the flare image, put it in the space of the 3D PI, and then you do a voxel-based image multiplication to create your flare star, and this is what you end up with. So in our research clinic, this is now being used routinely. Dr. Reich, every time he has a case to read, calls for our offline processing pipeline. These images are generated offline but pushed back into the PACS system, so all clinicians up in radiology can look at the flare star images. And it also been implemented on other sites. So University of Nottingham in the UK, University of Vermont has also this feature implemented and can do that routinely. So it's also giving us feedback from outside clinicians how useful this actually is. And moreover, we have used the NAMES network to actually propose a multi-center study around this. This is called the Central Vein Sign in the Early Diagnosis of Multiple Sclerosis, so CAVS-MS study. And because the PI is from Cleveland Clinic, that's why I think the CAVS term came in. Big fan of LeBron. And he managed to secure some funding for us and interest from 5 other sites. So there is Cleveland Clinic, Hopkins, UCSF in San Francisco, University of Toronto, University of Vermont, and University of Texas, Houston are now on board, have the protocol running on their site. And us, the NIH, will be the site for analysis of the image, and Penn will be for the statistic analysis. And so the goal is to slowly ramp up this multi-center, test it across the 6 sites with 10 patients per site, and see how stable and robust is the imaging, because maybe the way we do it in NIH will be different on the other side. So we want to make sure there's not too much variability and the clinicians are seeing the same thing across sites. And then, of course, if this happens that we have good image across different sites, then you can start asking the clinical questions. So for example, is GAD that important? Do we really need to do the GAD imaging? Or can we start testing the different criteria for the central vein? And then can we start comparing those with the existing criteria? So that, I think, kind of illustrates pretty well one of the goals of the lab, which is really to push the envelope on the imaging to help potentially change the practice in the clinic and provide better care to the patient, in this case, earlier diagnosis and earlier treatment. Now let me switch gears to another big topic in the lab, which is more about drug development. So all these great drugs that have been developed for the past decade or so really have been triggered by the development of a model, an animal model, which is, if you look at this timeline, happened actually fairly early in the 1930s. And this is quite a significant step for the industry of pharmacy drug therapies in MS. Because in this animal model, you can think about testing all kinds of drugs. And if you actually have a hit, then you can start thinking about moving to the human. So traditionally, EAE has been the main model for testing MS therapies for the past 20 years. And basically, it's a simple approach where you use an antigen, can be myelin-derived antigen, can be a myelin basic protein, can be MOG, can be all kind of different antigen you can think of related to myelin. And you inject it in the animal and wait for the immune system to respond. And then at some point you will see a neuroinflammation response into the CNS of the animal. And like I said, these rodent models, primarily the mice models, have been very successful in helping the field in finding new therapies. Unfortunately, for all the successes you have probably 100-fold failures. And this costs a lot of money, a lot of time. So there is a strong interest in trying to refine this animal model in something closer to the human that would actually help bring up the success rate to a higher level and put out novel drugs that are more helpful to the patient, especially the one I was talking to you that could help the SPMS or PPMS, or the more progressive patient. So neuroprotection, neurorepair would be, for example, a big application. And in our lab, and at the NIH in general, we have had a strong interest in non-human primate. And among those different primate, the New World monkey named common marmoset has been a strong interest for us. And for many reasons. First, they're tiny, so they're like a squirrel-like, so they're easy to handle for us in the lab. They have a high fecundity, so it's very easy to build up a colony from scratch. Our current colony is around 150 animals, and it was built about 10 years ago. So it's fairly quickly to ramp up a colony, and then you can think of many different applications, not only for immunity and autoimmune diseases, but you can also think about neuroscience, stem cell, all kind of application. Actually, Alfonso Silva, who's running the colony, will present a talk later on during the summer course, and we'll show you how you can use that to study brain functions using fMRI in this animal. But in our lab, it's really all about mimicking MS, so using this EA model as a bridge, basically, between the rodent world and the human world. So there's a great review from the Dutch group. First author is Cap, Yolanda Cap, and I highly recommend if you guys are interested in looking into that. But basically, in a nutshell, what it says is that the EA is actually being used in the marmoset back in the 95 by a fellow at the time called Luca Massacesi, who actually used human white matter homogenate. So there was not a very refined model, and injected with a complete adjuvant in the animal, and got a strong reaction. We actually had Luca come over visit our lab back in 2010, 11 to train us to do how to do this and refine a little bit more this induction. And we observe in our animal basically the same as what has been reported a few years ago, which is a strong demyelination, a strong inflammatory response in the brain and spinal cord of these animals. But the big strength of this model compared to the rodent is that you have now an imaging output, because the brain has a lot more white matter than the rodent do, and therefore you can better see the lesions. So at the time, here at the NIH, back when Harry McFarlane, the founder of our branch, started this program, they only had access to a 1.5 Tesla, and Joe Frank, who is now in the registry department, ran this program and was able to collect pretty nice images using basically a knee coil. So the human knee coil was placed on the head of the monkey, and they were able to collect fairly good images of the brain. And what they quickly see is that there was a close correlation between the number of lesions that appear in the brain and the clinical symptoms that the monkey were experienced. So what we did basically has been to re-update this approach using more modern scanners. So Dr. Silva had provided access to a scanner and resources and staff, and we've been able to really beef up our imaging on this animal. And we have an array of coils that have been built in Alfonso's lab, head coil, spinal cord coil. And we have all the tools that we need to monitor these animals, while they're being anesthetized and scanned for hours and hours. So this is what you can do when you have that kind of equipment. You can push the resolution in vivo to the limit of 150, 120 micron in plane, which really now starts showing all the details that you want to see in those brains and those lesions. So that's what we do currently on the imaging, but you can also start looking for contrast. So like we do on the human, on the 7T, 3T, and 1.5T, we usually have a battery of different sequences that highlight different aspects of the brain using different contrasts. And you can do that on a monkey. So we have here's our current multi-contrast battery that takes an hour. You can get all these different type of contrasts. And then you can also start thinking about using more advanced technique. This is a work done in collaboration with other staff scientists here across different institutes, where we basically try to highlight, for example, the white matter, gray matter contrast. We try to look with, I have a great grad students here sitting in the front, Nate, who's been helping me pushing also the imaging with what we call T2 star and phase. So we try to look here for iron accumulation, because those sequences are exquisitely sensitive to those particles. And then if you stick the monkey brain for a weekend, once it's been extracted from the body, obviously, you can actually get super duper high-res scans. And this is an example of a scan I did using diffusion tensor imaging. So you're looking basically at the fiber architecture of the brain. And what it shows is this monkey brain recapitulates all the main features of human brain. You have the journey of the corpus callosum, you have the splenium of the corpus callosum, you have the corticospinal tract, you have the optic radiation, anterior commissure. But of course, it's much simpler than the human brain is. And this is partly because this brain is encephalic. There is no circumvolution of the cortex, so it's a much simpler brain to navigate on MR. And I have also a terrific grad student in my lab, Jennifer, who's come from France, Paris, and with her we've been ramping up the spinal cord imaging in vivo. This is our latest image that we collected in vivo on these animals, the healthy guys so far. And this is what we see, a beautiful gray-white matter contrast all along the cord. So these are different levels of the cord. This is the top, this is the bottom of the cervical cord, and you can also go thoracic, and you have exquisite details as well. So now if you induce the disease in the animal, this is what you see on MR using those imaging technique. You start seeing those white spots like MS lesions. So they're bright and they distribute all around the white matter, different location. It could be affecting also the optic nerve, like MS patient have. It could be also affecting the gray matter. It could be also affecting the cerebellum. So he also recapitulates the radiological features that you see in MS by MR. And one other example of that is the central vein. So you remember how you see this dark line going through the MS lesion. So this is a 70 of an MS patient I scan here. You see the vein going through the lesion. And if you look at the same contrast I apply on the monkey, same finding. You see again this central vein, which really demonstrate the similar process happening here, where you have inflammation coming out of the blood vessel, leaking into the surrounding tissue and creating the damage. And then if you do inject this contrast agent, the gadolinium, you will see also the same feature, that the blood-brain barrier is open after the cytokines or whatever is being released from the blood vessel and attack tissue, and then you start seeing these lesions that are taking up the GAD, that tells you that they're acute and active. And you see this in human, but you see this as well in the monkey, using the same imaging. So the power of the imaging in the animal, of course, is that you control everything. You control the condition of the scanning, you control the timing of the scanning. Unlike patient, they are compliant, they come to the MRI, and they get everything done the way you want. So you can start doing this serial imaging very routinely in a very controlled manner. And this is what I'm going to show you here, is about a year of imaging summarized in 20 seconds, where you basically see the evolution of a time of these lesions that pop, grow, sometimes it might shrink, sometimes it might reappear. So that's the conventional contrast that usually is very sensitive but not very specific. We don't really know what's happening there besides the inflammation and demyelination probably there. This is a more specific advanced technique called MTR, magnetic decision transfer ratio, that a lot of people in the field, especially in the industry, pharma industry, believe it's actually very sensitive to myelin. So if you want to develop a drug that promotes remyelination, this is the kind of imaging you're going to want in your clinical trials. So we actually tested it in our animals, and I'm going to play it again. Instead of being bright on this contrast, the lesion is going to be dark, and you see the level of darkness, which is here really, really dark, tells you there's probably heavy tissue damage happening here, probably very important demyelination. So when you do that kind of study, you can start thinking like you do in clinical trials. You can start as having the same outcome as you will get in your human trials, which is an MR-based outcome. You can start counting the lesion, you can start assessing the volume of the lesion, you can counting the number of enhancing lesions. So your design of the animal trial becomes very similar to the human trial, which is totally new because in rodent, this is not feasible. So here's an example where we measure what we call the clinical score of the animal. So we have neurologists and fellows in the lab trained to recognize symptoms in the monkey, and they have a scale, and they, like you will get a neurological exam in the clinic, this is the same those monkeys get examined before the MRI, and we put a score. And we correlate with the number of lesions that we saw in the brain, and you see there is again a very tight correlation. Of course, those lesions that are in the brain are symptomatic and actually affecting the functioning of the animal. But unlike clinical trials, where you never have access to the actual tissue because humans are alive, the animals can be sacrificed at some point. And once you extract the brain, you can start thinking of all ways to cut the brain precisely to get down to the lesion that you saw by MR. And we have developed expertise in our lab through two very good students, Joe and Nick, who have been helping me really building up a 3D printing imaging program where we basically design our own brain holders and brain slicers so that we can carefully cut slab of tissue where we want to get the lesion. And there's a whole pipeline involved in this on MR side. So we have all the in vivo MRIs, and then we collect this very high resolution ex vivo MRI over the weekend to help us create these 3D models And then, thanks to the help of a very talented pathologist, Sankwan, we've been able to really get also beautiful staining. So now we can start addressing the big questions that a lot of MR physicists are asking is what my MR signal is related to in terms of biological change in the tissue. This is, for example, a way to answer these questions. So here's an example where we target lesions that we saw in MR, and you can see based on the stain on the top side, which is a luxofast blue, that all these lesions are actually demyelinated. Now, if you're more a neuroscientist and a physician scientist, you want to ask questions about the biology itself, and maybe think about ways to understand better the processing happening in the monkey brain. So a grad student who now is back in Europe, Pietro Maggi, he actually led a little study where he collected all the in vivo MRIs, did the histology, but then did the analysis backward. So he started from histopathology, finding that lesions that he see on the pathology, and then asked the question, how old this lesion is. So he went back in time, looked at all the MRIs that we had, and was able to determine the age, ballpark estimate, depending how frequently you image, but ballpark, few weeks, more or less, how old the lesion is. In that case, he found out that when you use weekly MRIs, you can define that this lesion is fairly young, is about 2 weeks or more, or less. So he started looking at more routinely in all lesions that he detected in his cohort of animals. And based on this large sampling of lesions, was able to start pulling out some groups of lesions per age. And he found out, for example, that in lesions that are less than a week old, those called acute lesions, there is a very characteristic pathological signature. Using a battery of different stain and immunostain, you can actually determine that those lesions are usually heavily demyelinated already. There is a heavy inflammation with macrophages and macroglia, and also a very dense immune response from some T cells and B cells, primarily T cells driven. And if you look at the lesions that are a bit older on MR, you will see a slightly different picture on the histology. So the lesions tend to be bigger because it's growing over time, tissue destruction is happening in terms of demyelination. There is also some axonal loss happening when you look at axonal stains. And there is still a dense response in inflammation, but you can see a slightly different pattern on the blood-derived macrophages that are now sitting at the edge of the lesion, whereas before they were sitting around the blood vessel, the central vein. And then if you start looking at older lesions, the one that persist and a little more chronic, then you see again another picture in terms of inflammation. So it's much less pronounced inflammation, but the demyelination is still there. And you can have still some axonal damage. Interestingly, when you start looking more carefully at the edge of the lesion, Pietro was thinking there is maybe some remyelination happening along the way. So that triggered our interest, and after he left, we kind of pushed a little more this direction of the work where we start looking at longer time periods. And that really depends on how long animals survive. And we've been having very heterogeneous disease course with animals lasting for a few months up to a year, and that's been by playing a little bit with the immunization protocol. And here's an example that an animal that actually lasted fairly long, so up to 54 weeks post-immunization. And what you can see is that a lesion in this animal appeared about 30 weeks post-immunization and lasted until the terminal MRI. However, the course of the lesion is quite interesting. It can keep growing until week 38 to 42. And you see it's brighter and bigger. But then as we pass this peak point, then it kind of starts disappearing progressively. And the terminal MRI, you would actually not even guess that there is a lesion. You still have a hint of it, but it's nothing compared to what you had at 38 weeks. Same with this little neighbor lesion. It's smaller, but kind of follow the same pattern. So if you start quantifying this and draw an MRI and look at the intensity in a more quantitative way, this is what you will see. So the intensity of lesion will start picking up, and then when it reaches peak, it will actually start dropping down back almost to its normal baseline intensity. So we're wondering if this is showing us any repair and actually showing us any remyelination. Again, remember, we have here access to the tissue after the experiment, and this is what we did. We used all our knowledge in terms of post-mortem imaging and cutting the brain carefully and getting down to the lesion that we're interested in. And this is a reminder of the lesion that was at the peak point where it was the biggest and the brightest. This is the terminal MRI in ex vivo conditions. And when you look on the staining for myelin, you can see that the myelin stain is actually quite there. It's nothing compared to what I showed you before, where you're completely absent. So that clearly indicates us that indeed what we're seeing is probably remyelination. And this is happening spontaneously because you're not treating the animal here. So from all this experience that we had in a few years, we started building this model for what happens into the marmoset brain when you induce EAE and you look at the lesion level. And we've characterized what we think are the main step of the process, with a perivascular inflammation starting early on, soluble factors being released, then you disrupt the blood-brain barrier that starts seeing the enhancement of the lesion, and then you have local activation of microglia. Up to that point, it's kind of hard to detect those by MR, and we've been looking hard, thoroughly through. It's really hard. I think it's beyond the resolution that we have. But once the lesion appear, then we can start following it and really tracking its evolution over time. And that's why we think, based on the pathology that we collected, we see the myelin damage happening, we see the inflammation going to the edge, and the lesion keep on growing and growing. And there is, we think, a myelin removal step that precedes, and it's been cropped out here, but I added a step which is spontaneous remyelination based on our most recent data. So if you are thinking about treating these lesions in the same way you would do MS, you have now a great model to test your drug. Because I think about addressing the inflammation part, but we already know that we have more than 15 drugs out there FDA approved for this. So maybe not the ideal first target. However, if you're into promoting remyelination, which should be shown here at the bottom of the text, this is a great model actually to address this and have an outcome that mimics the human outcome. So you can not only test your drug, but also validate the design of your trial. So when you go into human, you know not only that you have a good candidate on the therapeutic side, but you also have a good outcome on the MR side, which is really lacking right now in the field. So this is primarily brain disease, and if you look at MS patient MRIs, you will actually see there's a lot of spinal cord. And most of the symptoms that have been detected on neurological exam actually come from core lesions. So the core of MS patient is highly damaged. And this is what we can see as well in our animals, which is another strength of this approach. Is if you do MR of the cord in an EA animal, and this is the first lesion that Jennifer picked in vivo, we actually were able to detect it in vivo. Then we got the tissue out, look at ex vivo, and they actually confirmed that it was a lesion. And this is what Sangkwan did with the staining and really showed us that indeed it was completely myelinated. So we've been looking at that a little more extensively, looking in our bunch of tissue that we have in formalin and characterizing the postmortem, and we see this actually quite frequent like MS. So not only we could look at the brain MRI study, but we could also envision having a spinal cord combo where we can address the entire damage throughout the CNS using the drug. So, which is currently also a limitation. Human imaging of the spinal cord is very challenging, and having good imaging is always difficult, especially in clinical trials. So therefore, this could be, for example, a pre-clinical study where you look at the entire CNS. So you can look at the susceptibility of the lesion to repair more or less depending on the location of the CNS by imaging only. So this is how I'm going to wrap it up. I hope I was able to show you today through the research that is done in our lab how advanced MRI can help studying CNS disease. And I give you guys two examples, I think, which is first to help potentially improve the way MS is diagnosed, leading to earlier and more accurate diagnosis. And that was shown through this advanced technique that we developed here, T2-star weighting imaging using 3D API of TheraStar, and looking at the central vein as potential additional criteria for the diagnosis of MS. And then how you can also think about conducting preclinical research and testing novel therapies for MS using this quite advanced imaging that we have here from the marmoset, and also this fairly novel model, the marmoset EA. And of course, this is not my work, this is the work of a team. I've been blessed to work with very good fellows for the past 7 years, and a fantastic PI. Danny has a great vision for everything we do in our lab. And then, of course, we have strong collaboration with the other groups in other institutes, and also with partners in the industry, which are always looking for application of what we do, which is, I think, really the importance of the NIH is not only the basic science, but also putting back that into the daily life of patient with the support of people who actually do that. And I want to thank you for your attention, and I'm ready for any question you guys might have. Any questions? One question.
I have a question. So for the central vein positive CNS diseases you saw, so compared to MS, I saw that there are a number of other diseases that also has central vein signs. Is there any difference in terms of the size of the vein or the ratio or any different characteristics of the vein? Is it not actually not in the center versus?
So for the sake of the recording, I'm going to have to repeat your question. So the question is about the characteristic of the central vein in mimics of MS, right? Is there anything different related to MS lesion themselves? So this is not being investigated carefully yet. I think the field is still at the stage where we want to assess the difference in proportion, simply, just clinically, very simple way. But obviously there are tons of questions you can think about from the biology side. And one thing that always puzzled me is knowing the density of blood vessels in the brain. Statistically, there's a high chance that your lesion, whatever lesion is located, is going to hit a vein in through the center, right? And how come this we don't see in other conditions? We don't see veins. So you're right. Maybe the MR technique we use is not sensitive to those type of vessels. And why? Is it because it's too small? Is it because the amount of oxygen that creates a contrast inside the vein is different? Because what you see on imaging is not always what is the biology, right? There's the apparent size of the vessel, there's the real size of the vessel, there's also the source of the contrast that make the vessel visible. So there are many questions that we can ask from the biological side. This is something we're actually looking in our lab. Probably Martina and Govina actually try to go into more biology. But you can see the difficulty of this problem because if you want to address this question, you got to go post-mortem. And then, of course, everything is different. So maybe the monkey could be a way. So we've looked at monkey data, and what has been striking to us is you actually see the blood vessels before. They're normal, and then suddenly lesion appears. So there's not really any change in the EAE lesions in terms of central vein, which makes us think there's probably nothing really specific about it. But again, that's the model, so it's not MS. But I mean, really, it's an interesting point, and there should be more investigated in the future. Any other questions? It was so clear. Everybody got it. I achieved my goal. Well, I hope you guys enjoyed it, and for some of you I might see less next year. I hope I will bring you more interesting new stuff to show. Okay? And I'm going to wrap it up now.